Thank you for joining today's video. The topic is 6 different GPCR assays we'll go over and which to use and why for using for drug discovery development programs as well as research facilities. First of all, we want to talk about the biology of GPCRs. GPCRs, or G protein coupled receptors, remain the most prominent drug targets globally. There are about 800 plus GPCR targets, with 1/3 of all drugs to date targeting this family. With rhodopsin GPCR class being the largest family, roughly 660 receptors, this is a very diverse superfamily of drug targets. About 400 GPCR targets are active in diseases, and about half of these are currently undruggable, which is an excellent opportunity for researchers. And approximately 17% of those 400 are in drug trials today, or clinical trials. So these well-established targets are well known for decades of drug discovery development. They have key roles played in physiological processes such as visual, smell, taste, pain tolerance, etc., listed here on the slide, as well as many pathological roles: cancer, neuro, COPD, asthma, neurocrine, etc. So let's focus our attention to the top right-hand corner of this schematic image of a GPCR. It has the extracellular domain with its loops, the transmembrane region, as well as the intracellular domain. The ligands on drug targets obviously bind in the extracellular domain, and some actually insert themselves into the intracellular domain and change the conformation depending on the ligand that's being bound. Also in 2012, I wanted to mention there's a Nobel Prize that was received for studying GPCRs, specifically by Robert Lefkowitz and Brian Kobelko. So let's focus now on the different target classes. Sorry about that. The commonly studied families and targets are shown here. This data is based on publications, clinical trial data, and even scientific news articles. The ones in the table are the most commonly studied based on this information, but I did note other families that are popular like serotonin and the orphan receptors, bradykinin, etc. at the bottom. Focusing attention on the target families, there's glucagon. Glucagon has several different targets like the GLP-1 or glucagon-like peptide. They typically bind targets such as the peptides and the biologics. Most of the clinical trials using biologics, and research areas including metabolism, digestion, renal, obesity, etc. For the next two families, these are opioids and cannabinoids. The drug development process has been focused on small molecule type of drugs involved in CNS, neuro, pain, addiction, sleep, and hunger type of areas. For the fourth one is the chemokines. Lots of different targets for those, and mainly drug targets are the biologics focused on CNS, cancer, and HIV. And finally, we have the dopamine receptors. Several different targets for those, and most of the drugs target are small molecule focused and CNS, pain, addiction, and Parkinson's-like studies. So what are specific GPCR researchers working on when they're using GPCR assays? So current drug discovery programs from target ID, even basic researchers, all the way through QC lot release is shown here, focused on small molecule as well as biologics, which is the more growing type of molecule of interest these days. They're also looking at allosteric modulators, such as PAMs and NAMs, which are positive and negative allosteric modulators, as well as they're looking at applications focused on agonist type of studies. This includes partial agonists, as well as some antagonists. One thing to note also, crystal structures of GPCRs have been readily published in the last few years, and this is excellent because it's complementary to, or the in silico methods have been complementary to these in vitro type of methods or cell-based assays, which we'll be talking about. So if you look at the select applications for each area, you have your target identification and validation, which focuses on things like characterizing GPCRs, maybe looking at different isomers or isotypes, determining the mechanism of action of the therapeutic, de-orphanizing the receptors. These are, again, receptors that do not have ligands associated with them or known ligands. Maybe looking at multiple GPCR pathways. For instance, kind of like the ligand bias studies, you have a ligand that binds and either activates the cyclic AMP accumulation or calcium or beta-arrestin recruitment. So looking at that, maybe they mutate the GPCR, it causes a disease. They might be looking at that as well as internalization and trafficking. Then you move into screening and hit ID. Obviously, high throughput screening is still very popular, doing hit to lead studies, rank order, looking at potency and efficacy related to even structure activity relationships in the lead optimization step. Also, other orthogonal and secondary screenings mentioned in the screening section as well as in the lead op section for internalization studies. So we'll go over some of those assays in the next coming slide. But first, I'd like to step back a little bit and determine or show you what kind of product types are beneath or underlying these specific GPCR assays. So they're listed here. We have our stable cell lines. Stable cell lines definitely are utilized in many different labs. They have unlimited culture and they're functionally expressed. We have them available as separate frozen cell lines, in the liquid nitrogen phase, and they're used for all different types of applications listed on that previous slide. And then we have natural and traditional type of formats, which I'll go over a little more detail in a later slide on what this actually means. Then we also offer membrane preps. Membrane preps are popular for doing radioligand type of binding studies. These are more classic type of assays. And then also GTP gamma S, which are considered functional studies, but definitely using the membrane preps. Then we have our complete kits. These are called assay-ready kits. They come in two different flavors, express or bioassay. But in general, they contain the cells you need, one-time use, the plates, and the required reagents. And obviously applicable to all the different applications I mentioned on the last slide, including all the way to QC lot release, which is excellent for our bioassays. Next, we have some toolbox products. There's called DIY or do-it-yourself to allow customers like you to create your own GPCR cell-based assays. So maybe you need more flexibility than what the cell lines or kits offer. You want to evaluate your specific mutant or another species, an ortholog, such as monkey versus rat versus mouse. Compare it to the human version of that GPCR. Maybe you want to look at unique targets. So those are our toolbox products, and then we also have opportunities for services as well as custom assays. Custom assays are nice because we could provide you the cell pools, the cell lines, or even the assays. And then services let us run the assays for you. So our GPCR product portfolio mentioned all different types of product types, and I wanted to mention also that the assays cover a wide range of families. There are, I think it was about 80 different GPCR families, but this dendrogram to the right is showing you all the different targets, specifically in the A class, which is like the rhodopsin, and then the B and C class as well. So all the targets are around the circle or the phylogenetic tree or dendrogram, and then the little dots on the outside correspond to assays we have available for each of the target. If the dot is, say you wanted to have a binding assay for something that's missing, definitely contact our custom assay development and we will help you create an assay for that or give you the reason why it may not have worked. So overall, the coverage is about 90% of all the targets indicated in the human GPCR-OM, covering 290 unique targets. And the remaining, could be made through a toolbox, DIY type of approaches, or custom assay. So now we're going to move into the types of assays we have. There's about 1,600 functional and binding assays available. And these are the traditional type of force coupled as well as the naturally coupled functional assays. So at a high level, if you look to the left, you see our GPCR pathways where we have the biologic or the small molecule ligand that can bind. So you can look at binding, or you can look at functional type of assays, the signaling, either signaling through the G protein dependent pathway, or through the beta-arrestin recruitment pathway, or even other types of methodologies such as internalization and translocation, trafficking, etc. So to the right, just outlining a high level of what these assay types are. The receptor binding, looking at ligand binding, GTP gamma S functional. At the bottom, your G protein dependent type of second messenger assays, looking at accumulation of cyclic AMP or calcium, for instance. And then you have your other types of assays looking at beta-arrestin recruitment or receptor internalization. Now, to step back a little bit further about the technology that goes behind these assays, or most of them, is called our enzyme fragment complementation method. And this detection method is shown here on the right. Basically, it incorporates two recombinant beta-galactosidase fragments. This is the enzyme donor and the enzyme acceptor. The enzyme donor is obviously a lot smaller little peptide fragment that's unstructured, and it doesn't hinder the structure or biological interactions of the GPCR in any way. So once these two fragments come in close proximity together, they complement each other, and this forms an active beta-galactosidase enzyme, which, when the addition of a substrate, will allow the enzyme to hydrolyze the substrate and turn it into a chemiluminescent signal that can be read and give a nice dose response curve. Other types of benefits to this method is not only looking at a lot of different target classes, I'm meaning families like ion channels and kinases and checkpoint receptors, etc. So it allows you to do flexible, looking at multiple applications of GPCRs as well as other targets, allowing you to measure and rank ligand potencies, discover your mechanism action of your therapeutic, do binding and functional screens, etc. It is a simple homogeneous format, meaning there is no wash steps. It's a rapid mix and read type of assay format. So not only no washing, there's no centrifugation or filtration as well. And it's fairly fast, which I'll show you on the next slide. It's a robust method. It's an enzymatically amplified assay system, so it gives large signal to background ratio and high precision Z factors greater than about 0.7. Superior lot-to-lot reproducibility as well, especially needed for the bioassays. Overall, these assays have been well qualified and validated industry-wide, well established, been used for decades now. Essentially, they've been optimized for hundreds of different targets through screening and billions of data points, not only internally in our labs with our customers, but also our customers publishing and peer-reviewed publications. So we have thousands of publications around this technology, especially in the GPCR area. These assays are also scalable, so miniaturizable, from 96 going down to 3456 well plate format. We have examples of that. If you need help, you can contact our tech support for any more of this information. So let's move into the protocol. So in our GPCR assays, as well as other EFC-based assays, it's a plate format. Here you see your plate all the way to the left. You incubate the plate with the cells overnight, add your compound or biologic, incubate this for 1 to 3 hours, which is determined by the specific ligand or target of use, and that's indicated on the data sheets for the product. You add your detection reagent, which has the substrate, and incubate this for 1 hour in the dark, and then read it with any, assay plate reader. Just a standard luminescent reader is fine. And then you give a chemiluminescent signal, and then you're able to create your dose response curve. I already mentioned some of these points down at the bottom, so I won't go over those any further. All right, let's move into the assay types specifically for GPCRs. Okay, so we have about 6 ones we're going to cover. The first 2 are second messenger accumulation type of assays focused on cyclic AMP and calcium. And here's kind of the pathway going down, the activation of a G protein, decoupling from the beta and gamma subunits. The alpha subunit either goes off and activates the phospholipase C down to the pathway of N-acetyl triphosphate to calcium, or activating the adenylyl cyclase enzyme that converts ATP into cyclic AMP. So this pathway is GPCR dependent. It's an amplified system that leads to rapid saturation using low levels of your ligand. More sensitive system based on partial receptor occupancy, giving a full signal due to the amplified signaling event, especially after the ligand activation. So you can monitor the functional status of your GPCR directly by quantifying the cellular cyclic AMP accumulation levels in a dose-response manner. You can characterize your ligand pharmacology, with precision, and evaluate ligand bias. And this is nice because you can complement it with other types of assays that we'll talk about specifically, for instance, the β-arrestin assay on the next slide. Excuse me. These second messenger assays are excellent for looking or evaluating agonists, and it's because they can be easily detected. It doesn't take much agonist to obtain a full signal. But this is not necessarily true in an antagonist mode, where we recommend using the next assay, the β-arrestin one, instead, as well as partial agonists, using the β-arrestin first. The performance is reproducible, and it has all the lovely characteristics of the EFC technology, such as large assay windows and broad sensitivity ranges, and very specific to the target of interest. The format of these are chemiluminescent or fluorescent. Chemiluminescent uses that EFC technology. Fluorescent would be using an HTRF type of readout. Excuse me. Cell lines and frozen cells are available for these, and I wanted to talk a little bit about the traditional versus newer naturally coupled receptors. The force-coupled receptors are very popular and commonly used, throughout the decades. It is based on an overexpression system that increases the G-alpha specific subunit, accumulating to drive the signaling process, measuring the calcium or cyclic AMP. Unlike the other ones, these are naturally coupled receptors. They're ideal and more native or closer to the true biology for directly measuring or monitoring the activation status with high confidence. So, no overexpression of a specific G-alpha subunit that's not more natural to the GPCR that you're focused on. All right, and then we also have the calcium flux assays to measure calcium mobilization or flux in the cells based on activation status of the GPCR. These are cell lines or frozen cells, and they're qualified to use with either Flipper or any of the dye-based assays available in the market. And then they have all the same qualities I've mentioned before as far as large assay window, broad sensitivity. And also available in the native state with the GQ coupled state, as well as the forced coupled, the non-GQ state. All right, the next assay to talk about is the β-arrestin functional assays. These are universal GPCR assays, and what I mean by that is they're not dependent on G proteins. They're ideal system for discovering antagonists, looking at partial agonists, GPCR deorphanization, when you don't even know, the coupling or anything about the GPCR pathway itself for your specific target, dissecting ligands in general. So we highly recommend to use this type of assay first and complement it with a secondary messenger type of assay. So this G protein independent is a non-amplified system, meaning it's like stoichiometric. It's a one receptor for one ligand type of interaction. And then what happens after that is once the ligand is bind, the GPCR is actually internalized, leaving it absent and not available for additional ligand to be bound. So over time, the GPCR does recycle and become available. This method requires full occupancy to the ligand to be bound to give a full signal. Thus, you can study virtually any GPCR, which is really nice. I mentioned it down here, despite the different coupling status. It is a less sensitive approach, though, for agonists. It's more sensitive for antagonists that are too weak to be seen in second messenger systems, and it is good for partial agonists as well. So that's down here. Currently, this assay is only available as a chemiluminescent readout. All right, the next assay is our GPCR internalization. It's kind of similar to the β-arrestin one, at least one of the flavors of or formats of this assay. Like most of the other assays mentioned so far, the GPCR internalization assays are based on the EFC technology. This specific assay provides a tool for secondary or orthogonal screening to identify safer drugs, so drugs that give less undesirable effects such as drug tolerance, unwanted side effects, and diseases. So, you measure your GPCR in live cells and you have the ability to quantify this and gain a signal type of chemiluminescent readout. So basically quantifying internalized GPCRs localized to intracellular endosomes is shown here. So you can monitor the activity or the activated GPCR in live cells without expensive, microscopes, imaging equipment, or bulky labels and radioactivity or antibodies. So this is kind of a nice alternative. It just uses standard instrumentation, luminescent reader, no complex data analysis or normalization or pit vesicle type of spot detection is required. It does come in two formats. You're seeing one of the formats to the right here, where it looks at total internalization is a combination of looking at the GPCR where it gets endosized host into the cell based on normal biological mechanisms, or bound to a ligand it gets internalized. Or there's an activated internalization assay which looks at ligand-induced beta-arrestin mediated receptor internalization. So it's a lot like, obviously, that previous assay I mentioned. We got 100 of these cell-based assays that definitely uses the EFC readout like I mentioned before. Next is kind of a unique flavor of our GPCRs. I'll have a video for this available for you. It's looking at pharmacotrafficking assays to identify and optimize pharmacological chaperones. And what these are these proteins, these small molecules that promote proper folding and assist in the trafficking of the GPCR to its intended location, which is the membrane. As well as you can monitor this to go internalize as well. So the pharmacochaperone must be able to penetrate through the cell membrane and bind to the specific GPCR and correct it or stabilize the specific misfolded receptor or mutated one and allow that to be pushed all the way through to the membrane where it can function properly. So you're basically doing like a functional rescue, like a relevant type of assay, and you're focused on maybe mutations that cause diseases such as CTRF. Cystic fibrosis. Another application to this is evaluating incorrectly assembled receptors or ion channels or transporters. So this can be used as well. This uses the EFC technology, so you have all the wonderful benefits of that technology, and you can look at detecting antagonists, agonists, and allosteric modulators just like the other ones. And what's nice about these as well is like the arrestin ones, we do have toolbox products for you, so you can create your own pharmacotrafficking type of assays, just like the arrestin ones, etc. And then finally, we have our membrane preps. These membrane preps are used for doing radioligand binding and GTP gamma functional assay type of studies. They're single use, just like our assay ready kits, high quality membrane preps to identify ligands that bind to your specific GPCR. So you'll be looking at things like saturation and a competitive type of analysis with your radioligand or GTP gamma S functional based assays. So either one of those, they're highly reproducible results, high total binding, etc. But it is a radiometric readout type of assay. Overall, use all these multiple different assay types to have a full, complete understanding of your GPCR and therapeutic or ligands of interest. And before I in this, I wanted to bring up there is detection reagents or kits available. Some of the detection kits are required for these cell lines, for instance, and are coupled together inside of the kit. They come with it. So the Pathfinder detection kit is required for the β-arrestin recruitment, pharmacotrafficking, and receptor internalization GPCR assays. And then we have two other assay types for the cyclic AMP and calcium readouts. These are hit hunter assays. These specific assays can be used with any cells that have the GPCR in them. So not just Eurofins DiscoverX, but your favorite GPCR cell lines. All right. So choosing the best solution to meet your GPCR assay needs, first of all, you're going to want to know, what type of GPCR you're focusing on, whether it's a family, a specific target. Is it a human version, or maybe you're looking at orphan receptors or another species, such as orthologs, rat, mouse, etc. You're going to want to determine whether you're going to focus on functional or binding or both. You want to know the different assay types, cyclic AMP, GTP gamma S, internalization, whatever you're focused on. Then you want to determine, am I looking at agonists, antagonists, or both, looking at some kind of a ligand bias study, looking at PAMs and NAMs for allosteric modulations. Maybe you're focused at the end stage of your clinical trials and potency, or preclinical trials, potency, lot release type of studies, or focused on the mechanism of action of your specific therapeutic. Solutions are summarized at the bottom. We can do, quick testing using the assay ready kits. These are the Express and bioassay kits that are ready to use, 96-well format and smaller scales. You can screen in-house, for instance, using the stable cell lines and membrane preps, or have us develop the custom assay for you, and we can deliver that in the form of cell lines or assays or cell pools. Let us do the profiling and screening for you through our services. Or you can build your own assays with our toolbox products and vectors and parental cell lines and starter kits. Other resources, not just technical support, can help you. You can go to our website, discoverx.com/GPCRs to access our product brochure with a similar information as indicated in this video. We also have a product solution guide, which is nice. It actually lists all the different GPCR products, and it's updated monthly. We have a beautiful GPCR wall poster as well. You can hang in your lab, the human GPCR own, with that large dendrogram, and then a family and target table at the bottom with the different coupling status of your specific targets. We also have reference guides with all the different dose response curves specific to all the targets, so we have that available. Publications and reference, endless ones, for GPCRs. If you want a specific one, don't hesitate to contact us and we can find you one for your specific target and application. We do have posters and presentations and many blogs and application notes, case studies and white papers, and even webinars for you to get further information on all the topics that I've discussed today. So with that, I'd like to end by summarizing the video we went over, talking about all our different cell-based assays, the functional and binding ones. We talked about the different assays as far as traditional force-coupled ones versus newer naturally coupled ones, based on our EFC technology. And then we have mentioned that we do have a large coverage approximately 90% of the GPCR ohm, the human one, covering about 290 different unique targets. Unique offerings, you just try our ready-to-use assay kits to get you, confident on the assay, and then consider a cell line or a custom assay, or even a more flexible option such as our DIY or do-it-yourself type of products. These are the toolbox products, vectors, etc. And then obviously access all the different publications to learn more about how different other researchers like you have published on or that will focus on different targets and assays. There's probably over about 900 now peer-reviewed publications for GPCRs since the early 2000s. So with that, I'd like to thank you for listening and look forward to hearing from you. Thank you.